Wait.
I'm gonna have to take that out a lot. I have to like 4 or 5. It was like one said like shitty comments. It was I can't believe that hurts. And then the one said I think I'm good at Smash. Right? I had to put like on his mirror, his window, like under his door handle, and shit like that.
Okay, let me reinforce something. I was looking at some of the little lysine, little small plastic tubes, the 4-hour ones you were supposed to look at, and we're getting reactions, but some of the controls aren't behaving the way that we had expected them to behave. In a lot of cases I looked at, it looked like both of the controls that I gave you were giving a positive reaction. But then I looked through other tubes, and some people's unknowns were giving a negative reaction. So the test is working. What's happening is that these bacteria, with their short generation time and their high tendency to mutate, can over time form new strains. And so, you know, we might have a strain of Proteus vulgaris, for example, which is supposed to be, you know, lysine negative, which has mutated to a lysine positive strain. So really, all you can do is you can take your pictures, and then I'll kind of keep an eye on these tests and make sure the tests are working. But sometimes we have mutant strains, and we just have to deal with that. I've had a couple people ask about your skin and throat and nose. You're getting results that indicate Staph aureus. It's catalase positive. It's growing on MSA. It's fermenting mannitol, but it's not beta hemolytic. You know, you probably have a non-beta hemolytic strain of Staph aureus. So in the microbial world, that does happen, and we've just got to kind of live with it. That makes it a little frustrating in terms of identification, because if you're going through a key and you hit a point and there's a test and your strain is a mutant for that test, it could lead you into the wrong place in the key. Okay? Well, when you start doing the keying process, one of the things you're going to find out is when it starts leading you to the wrong place in the key, you will eventually figure that out because you'll start reading the genus and species description of what the key says it was, and you'll go, that doesn't match at all. And so sometimes you have to go back and kind of try other alternative pathways, saying maybe this test didn't work out the way it should. So your grade on the unknown part of the project, it's not just all wrong or right. You know, if you didn't get what I said I gave you, you get no points. It's more about the process, and sometimes you'll get full credit, even though you've identified it as being something different than what I gave you, because that's the way it keyed. And so one of the things I check for is, did you key it properly? And then one of the reasons I have you take pictures is, did you interpret the results of your test properly? So if you looked at something and it was purple and you called it gram-negative, well, that's a problem, okay, because you misinterpreted a test result. But if you interpreted the test result properly and your particular strain that we're working with was positive for a test and the key says it's negative, that could lead you astray. Okay, so don't get too overly worried when you start working on your unknown about just getting the quote-unquote right answer. It's more about the process of things than it is the right answer. Now, hopefully most of the time you get to the genus without too much problem. But once you start getting down to species, strains do develop. Because I've been maintaining these cultures now for 4 or 5 years. And so sometimes mutant strains do develop, and you've now got a mutant strain. And I take that into account when I do the evaluation of your unknown. Okay? So just a kind of a word of comfort, if nothing else. So tomorrow we'll look at our results from Wednesday and do some more recording and documentation. Next week we're going to do another little project with some differential media, and your unknown will be inoculated on some of that media. That will just give you some backup information. But right now you've got most of the stuff you need where you can start working through that genus key to try to figure out what your genus is. And then remember, once you figure out what the genus is, you go to Bergey's manual starting with the 7th edition and use that to figure out what your species is. Okay, so we'll talk a little bit more about unknown identification Wednesday next week in lab. Okay? So, what does hemopoiesis refer to? Blood cell development. Okay? What are the two major categories of white blood cells that are produced during hemopoiesis? There are the two major types of white blood cells. Granulocytes and agranulocytes. Granulocytes have what? They have these prominent cytoplasmic granules which stain different colors during the right staining process. And agranulocytes have tiny, almost invisible granules. What are those granules? Yeah, they're various storage kind of compartments within the cell, one of the principal ones being lysosomes. Okay? Which are more numerous, granulocytes or agranulocytes? Granulocytes are more numerous. What's the approximate breakdown? About 2/3 of the white blood cells are granulocytes and about 1/3 are agranulocytes. Now, the ranges for those values are quite wide, and that's just an approximation. I mean, if you look up here on this particular slide, it talks about the prevalence and circulation of the granulocyte neutrophil, which is the one we were talking about last time when class ended. And look at that range, 55 to 90%. Well, the number of neutrophils that you have will vary dependent upon, number one, do you have an infection? Because there can be a dramatic increase in certain cell types if your immune system is stimulated as a result of an infection. Some of the chemical signaling compounds that are produced during inflammation, which we're going to talk about pretty soon, can step up production of certain types of white blood cells. So these numbers right here, the ranges can be fairly big, but that 2/3, 1/3 is just a ballpark kind of relative comparison to give you an idea of relative abundance of those two types of cells. Remember that the right stain was a differential stain, and that the two primary stains in there, one was a positive basic stain, methylene blue, and the other was an acidic basic stain, acidic stain, negatively charged eosin, which was a red stain. Neutrophils during the right stain are lavender in color because their cytoplasmic granules are taking up those components of the right stain. And they're lavender because those granules take up both acidic and basic portions of the stain. These particular cells are one of the larger components of white blood cells, and their primary role is general phagocytosis. And we're going to talk about the process of phagocytosis in a little bit. Relatively short lifespan, only lasts a couple of days. And only spend a little bit of that time moving around in the circulatory system. These particular cells, remember, are produced in the bone marrow and are released from the bone marrow following hemopoiesis and circulate around. But pretty quickly on, they migrate out of the circulatory system into the surrounding tissues. That's going to happen in response to infectious agents. So we'll talk about that process of diapetasis and phagocytosis in a little bit more detail. One of the things that characterizes them is that they tend to be a very multi-lobed nuclei. And so they have a pretty distinct appearance when you look at them under the right staining process. And I'll show you another slide of that in a minute. A couple of other types of granulocytic cells. Let's hop down here to this third one. Basophils. Basophils are granulocytic cells that take up the basic portion of the right stain only. So the composition of their granules is a little bit different, and that's reflected by the fact that they take up different kinds of stains. You'll notice in the neutrophils that a lot of the things that are found in these cytoplasmic granules are digestive enzymes. If you look at basophils, the granules contain some different kind of compounds typically. They contain histamines and prostaglandins and other chemical mediators of the allergic response. So these particular cells are heavily involved in the process of inflammation. And inflammation is part of your second line of defense, and it's a generalized response to both infection and injury. So why is it saying allergy? Well, allergies are actually an inflammatory response. And the symptoms that you suffer if you suffer from allergy are the result of these inflammatory mediators like histamine and prostaglandins being released by cells. How does that differ from inflammation? Allergy happens in response to things that are harmless, like pollen, and that are triggering an inappropriate inflammatory response. True inflammation is responding to infection and injury. So if you suffer from allergies, you are suffering from an inappropriate inflammatory response to things that the body shouldn't be responding to. So what can you do to treat some of the symptoms of allergy? You can take drugs that counteract the effect of histamines, antihistamines. And you can take drugs that counteract the effect of prostaglandins. You can take anti-prostaglandin drugs. So allergy medications tend to target these substances that are produced by the basophils that are responsible for producing the symptoms because they're inflammatory cells. There is a kind of a cousin to basophils called mast cells. Where basophils can migrate around in the body, mast cells are fixed in position. And you find a lot of mast cells associated with connective tissue. Under the proper stimulus, both mast cells and basophils can degranulate. Their granules break open. And during degranulation, some of those substances that were in the granules, like histamines, are released. And those are responsible then for inflammation and allergy. So mast cell degranulation is an important event in both inflammation and allergy. Not many basophils. You see that there are the smallest percentage of all of the granulocytic cells. And so, if nothing else in terms of kind of keeping track of numbers, I think maybe rank order is important. So knowing that neutrophils are the most common, then eosinophils far less, but more than basophils. Don't worry about absolute numbers for percentages. Other than that, 2/3, 1/3 approximation. Okay, eosinophils. What are they? Well, we're not absolutely sure what they do. There are some pretty good guesses. We think that their primary function is the destruction of large eukaryotic pathogens, like some of the multicellular helminthic worms or various fungi. We know that the number of eosinophils increases dramatically as a result of these particular type of infections. One of the places that you tend to find eosinophils is in the spleen. Remember what function the spleen has. The spleen is kind of a filtering device for the circulatory system. And so if you get a parasitic infection of the blood or a fungal infection of the blood, as that blood passes through the spleen, it will encounter these eosinophils which will respond to it. Their granules contain a number of toxic proteins which can kill the worms and the fungi. They also, though, contain various inflammatory mediators like the basophils do. So they do play some role in inflammation as well. The last two cell types on this particular chart are the agranulocytes, sorry. So these are the ones that have the less lobed nuclei, and they don't have the prominent cytoplasmic granules. So they appear different when you look at them in the right state. Let's take a look at monocytes first. Monocytes are the precursors to macrophages. So during hemopoiesis, the cell type that is formed as a result of differentiation is a monocyte, but then that cell will further develop and change its nature and turn into a macrophage. And this occurs under the influence of various chemical mediators, many of which are produced during the process of inflammation. So inflammation kind of stimulates this development of monocytes into macrophages. Initially, when a stem cell divides in the bone marrow, it produces a fairly small cell called a promonocyte, which then is released into the circulatory system. This can move around in the circulatory system for up to a couple of days. And then those monocytes will eventually migrate out into the tissues and complete their development into macrophages. And that happens more readily if they receive the proper chemical signals, like the signals produced during inflammation. One of the things that happens to monocytes is that they increase in size pretty significantly as they develop into a macrophage. There's about a 5 to 10 times size increase between a monocyte and a macrophage. Now, we've already talked about macrophages. Macrophages are involved in phagocytosis, but remember they are kind of the officers of the immune system compared to the neutrophils, because in addition to being able to just ingest things, they're antigen-presenting cells, so they can show the antigens to other immune cells. Oftentimes that occurs within the lymph nodes. And macrophages also produce a number of chemical signaling molecules, lymphokines, which help aid in the development of the rest of the immune response. A close relative of macrophages are dendritic cells, and we'll talk about those in just a second. There's one other thing I want to tell you about macrophages first. Macrophages, when they get in the tissues, tend to be found in two locations. Tend to have two kind of roles, I guess maybe I should say. Some of them are fixed within particular tissues. So these macrophages migrate out and become localized in a particular area. And that's where they spend the remainder of their life. Some of them, though, continue to wander around and patrol around, particularly through the RES system. So there are those wandering macrophages moving through the RES system, groping the other cells, and checking them out to see do you belong or do you not belong. The macrophages in the fixed tissues are typically located at common sites of entry, which can be common sites of infection. So the skin, the lungs, the liver, all have their own resident macrophage populations, and each one of those populations is given a different name. So for example, here are some alveolar macrophages. So these are associated with the respiratory system, which is one of the major sites of entry for pathogens into your body. Okay, so these fixed macrophages tend to be localized in those particular regions and to eliminate pretty quickly pathogens that enter through the respiratory system. There are populations of fixed macrophages in the liver as well. Instead of being called alveolar macrophages, those are called Kupffer cells. And each one of these different types of macrophages has a little bit different appearance and some adaptations associated with their particular environment. Langerhans cells are a type of fixed macrophage as well. They tend to be located in the skin, in that boundary between the dermis and the epidermis. This is a good summary slide for a number of reasons. It kind of shows you the overall process of what's happening during hemopoiesis. But additionally, it shows you the major players and how they differentiate from one another visibly based upon right-staining reactions. So if you look at a stem cell, hemopoietic stem cell in the bone marrow, you can see that there are several different lines of differentiation. There's a line of differentiation that leads to formation of red blood cell. There's a line of differentiation that leads to platelets. And then there are several lines of differentiation which lead to the various white blood cells that we talked about. And I didn't expect you to remember all of these names of lines of differentiation. There's a myoblast line of differentiation that gives rise to the granulocytes. And you can see that that particular cell can differentiate along this pathway and then also can give rise to the mast cells. So the mast cells and the neutrophils, basophils, even though they have different functions, different structures a little bit, proceed along different lines of differentiation, but these are the granulocytic cells. And then there are numerous lines of agranulocytic differentiation as well. So there's one that leads to the differentiation of the monocytes. The monocytes give rise to the macrophages. And you'll notice that the precursor to macrophages also gives rise to kind of the cousin of the macrophage, the dendritic cell. Dendritic cells and macrophages functionally are pretty much the same type of cell. You really can consider this to be kind of a cousin to the macrophage. The main difference is dendritic cells tend to be localized more in epithelial tissue and particularly in the skin. But they are able to be antigen-presenting cells. They are able to produce lymphokines and other chemical mediators. So functionally, they have a lot of the same roles. So we have talked about this line of differentiation a little bit, and I need to fill in a few gaps. There is also a line of differentiation that leads to lymphoblasts. and lymphoblasts differentiate along two lines. One is the T's, and then it looks like they've got a typo in it. This should be B's. So there's a T line of differentiation and a B line of differentiation. T lines lead to T cells, B lines of differentiation lead to B cells. And again, it should be starting to become kind of second nature now. B lymphocytes associated with humoral or cell mediated. Humoral antibody production. Okay? What's the type of B lymphocyte that actually produces the antibody? That's the plasma cell. Okay? T line of differentiation leads to cell-mediated immunity. Three types of T cells that we talked about. Sarah, what were they?
Helper T cells, killer T cells, and suppressor.
Helper, killer, and suppressor T cells. That's something we asked for in our senior or your seminar. What do T helper cells do? They help B lymphocytes produce antibody, helping the B lymphocyte produce antibody through some chemical signaling. Where are B cells produced and educated? Bone marrow. Okay. Where are T cells produced? Where are they educated? In the thymus. And what does education involve? Building yourself a particular receptor for a particular antigen. So eventually we're going to come back and talk about that gene shuffling that occurs that allows all these different cells to produce unique receptors for particular antigens. B and T cells are involved in the third line of defense, which is the specific line of defense. That is the acquired level of immunity, as opposed to the innate lines of immunity, the first and second line of defense. So this is where you get specific antibody production and killing of specific cells that have become damaged either by becoming cancerous or virus infected. Now, there's this other little line of differentiation over here in the lymphoblast differentiation pathway. You've got B and T, and then you've got this other weird cell down here. That's called a natural killer cell. Functionally, natural killer cells are a lot like T killers. How are they alike? Both of them kill damaged host cells by punching holes in the membrane of that cell. There's a system we'll show you when we talk about cell-mediated immunity. T killer cells are part of the acquired specific immune response. And the only way a T killer cell is activated is in response to a particular antigen. Natural killer cells don't have that level of specificity. They instead kind of generally kill cells that appear to be damaged, but they're not stimulated by that specific antigen signal that T killer cells are. So it's kind of a more generalized response to bad cells. And we'll talk a little bit biochemically about how those differ when we get into some more detail about this. But you'll hear reference to T killer cells as we go on. Down at the bottom is a nice right-stained set of slides that show you the appearance of these particular cells and the difference in their nuclei and the different colors of the cytoplasm of the granulocytic cells. If you have a blood sample set off by your physician to a medical technology lab, one of the things that the MedTech lab will do is do a differential blood cell count. And that will determine how many red cells do you have, how many platelets do you have, how many white blood cells you have, and what type of white blood cells you have. Now, this is becoming more and more automated. But one of the things you probably still do in medtech programs is you have to microscopically be able to look at a right stain slide and to be able to count how many neutrophils are there. We have to be able to tell what a neutrophil is. How many eosinophils are there? And this can provide you some interesting information. Number one, what does it indicate if the general number of all white blood cells is elevated? Well, that might indicate something like a leukemia, for example. Okay, and the bone marrow is cranking out many more white blood cells than it should. What if you get a certain type of white blood cell elevated relative to the rest? That can be diagnostic in terms of particular types of diseases. There is a viral infection that tends to dramatically elevate the number of monocytes in your blood. And a differential blood cell count will point that out. What's that disease? That is mononucleosis. Okay, that's where the term mono comes from. It's an elevation in the number of white monocytes. So you can gain some interesting insights into infection and diagnosis of disease by doing that differential cell count and seeing what your relative number of white blood cells are. Well, there are some unique characteristics of white blood cells that are really important. Because a lot of white blood cells are involved in phagocytosis. Phagocytosis occurs within the tissues. That's primarily where the pathogens are gobbled up. But the white blood cells are originally released by the bone marrow into the circulatory system. So one of the things white blood cells have to be able to do is they have to be able to move from circulation into the surrounding tissues. And that is that phenomena that I mentioned earlier, diapetesis. Or leukocyte extravasation. So let's take a look at diapetesis for a second. How do those cells get out of the circulatory system and into the surrounding tissues? Okay, so here's the inside of a little vessel. This probably is a little small capillary. Capillaries tend to have single layers of endothelial cells making up the outside of the vessel. And then here's the lumen where the blood is flowing. And you'll see that in this particular situation there are a number of neutrophils. Well, why are there a lot of neutrophils in blood? Well, maybe the bone marrow has been signaled by the inflammatory system. There's an infection, let's start producing more of these particular types of white blood cells. But they're released into general circulation from the bone marrow. How do they get to the specific site of infection? Well, typically there is some type of chemotaxis involved, an attraction to a chemical stimulant. Those chemotactic substances can be a product of or part of the pathogen itself. So LPS can be a chemotactic substance. Peptidoglycan can be a chemotactic substance. There are immune cells which can release chemical substances which are also chemotactic. Lymphokines. Inflammation, if it's triggered, can release chemotactic substances as well. So at the site of infection, we tend to get an accumulation of substances which will attract those phagocytic white blood cells, like the neutrophils. Diapetesis then refers to the actual migration of the white blood cell from the interior of the circulatory system into the surrounding tissue fluid, where the pathogen is most likely located. Now, there's some interesting biochemical stuff that's happening both within the endothelial cells of the vessels and within the white blood cells themselves that make this diapetesis possible. So here's another diagram. This one's a little bit harder to see. So here's our vessel right here. Here's our endothelial cells right here. Here's our white blood cells. And eventually these white blood cells, as they are moving through the circulatory system, will start to adhere to the walls of the small vessel. They'll stick to the endothelial cells, and then eventually they will push their way out through the wall in between the spaces of the endothelial cells and migrate into the surrounding tissues where the pathogen is located. There's some interesting chemical signaling that takes place under the response of various signaling molecules, many of which are produced by inflammation or other immune cells. There are changes in both molecules called selectins within the white blood cell and molecules called adhesins that are in the endothelial cells. And different genes start getting turned on, and different molecules start being produced, so that some of these selectins in these cells, for example, bind to the adhesins in these cells, and that results in the white blood cell getting kind of stuck, so it can then migrate or extravasate out into the surrounding tissues. So there's some really interesting changes in gene expression and production of new proteins, dramatic changes within the organization and structure of the white blood cell itself that allow it to change shape and to migrate through that narrow little passageway. Once you're out in the surrounding tissues, then the white blood cell can engulf and internalize a pathogen like a bacterium. So this is a neutrophil now in the process of phagocytizing a bacillus-shaped bacterium. The process of phagocytosis is a pretty complicated process. There are several steps that typically are associated with phagocytosis. Let's take a look at what some of these are and what's involved in this particular process. So, here's our neutrophil up here in purple. It is migrated out of the circulatory system in response to chemotactic factors produced by these pathogens or by inflammation that has been stimulated by these pathogens. So something is attracting this particular neutrophil. The first thing that has to happen after chemotaxis is we have to have an attachment occurring between the phagocytic cell and the pathogen that's going to be engulfed. No attachment, no phagocytosis. So that's why capsules, for example, tend to make certain strains of bacteria more virulent, because they interfere with that attachment phase. They cover up the surface of the pathogen and make it difficult for the receptors to recognize the PAMPs on the surface of the pathogen. Remember what PAMPs are? Pathogen-associated molecular patterns. Molecules that tend to be pretty exclusively associated with bacteria and other pathogens. Peptidoglycan, flagellum, those are common PAMPs that the receptors on the surface of the phagocyte can recognize. One class of receptors on the surface of the phagocytic cell that have been studied intensively are the toll-like receptors. So toll-like receptors are receptors on the surface of phagocytic cells that recognize molecular patterns on pathogens and allow binding behavior. So here is a macrophage showing these toll-like receptors on its cell surface. Here is a binding of a foreign molecule, a PAMP, to that toll-like receptor. And typically what happens is when the toll-like receptor binds a PAMP, a signaling cascade is activated, which turn on some genes in the nucleus of that cell and result in the cell now producing various chemical molecules, cytokines, interleukins, inflammation compounds, which are going to be responsible for other aspects of stimulation of the immune system. Some of those might, for example, attract additional phagocytic cells. Some might stimulate the formation of B lymphocytes into plasma cells so antibodies can start being produced. Here is a list of some of the currently identified toll-like receptors, and I don't expect you to memorize what all these are. You'll notice that there are 10 of those that have been identified to date, and that they occur in various portions of the cell. Several of them occur in the cytoplasmic membrane of the phagocyte. Some of them, however, occur in the phagosome membrane. That's a term you should be familiar with. You probably are. When ingestion takes place, so when these pathogens are ingested, eventually they are enclosed within a vacuole. And that vacuole in which they are enclosed is called a phagosome. Eventually, that phagosome will fuse with a lysosome. So now the pathogen is exposed to all those killing compounds in the lysosome. That's called a phagolysosome. So there are some toll-like receptors associated with the phagosome membrane as well as the plasma membrane. And then there's one that they list here that has unknown location. I think the interesting thing about this particular slide is what are the PAMPs in this particular situation that the toll-like receptors are recognizing. So this gives you kind of an idea of what kind of things are PAMPs. Lipid A right here, okay, part of the LPS layer. Okay, lipoteichoic acid, that's part of peptidoglycan, for example. Double-stranded RNA, found only in certain types of viruses. Okay, flagellum. So these PAMPs are molecules that typically are not found in host eukaryotic cells, but are typically found in pathogenic microorganisms. And these toll-like receptors have learned to recognize these general classes of molecules and bind to cells that possess those, and then internalize them and destroy them. So this is an important part of your second line of defense, phagocytosis. Okay, so once you're internalized and inside of a phagosome, then the next step that's typically involved is phagolysosome formation. So the phagosome fuses with the lysosome, and now the pathogens are exposed to this kind of toxic brew of chemicals, some of which include enzymes like lysozyme. Remember what lysozyme does? Degrades cell walls, breaks down peptidoglycan. What is DNase going to do? Break down the DNA into nucleotides. Okay, RNase, proteases. And then also remember, within that phagolysosome, we get what we referred to in the last exam as a respiratory burst. And the phagocytic cell starts taking up oxygen very dramatically and starts metabolizing very rapidly. And what does it do when it starts metabolizing aerobically very rapidly? It starts producing all of these ROS compounds, superoxide anions, hydrogen peroxide, singlet oxygen, hydroxyl radicals, hypochlorites. Hypochlorites, remember we talked about in the context of bleach. So a lot of these ROSs are produced, which help the digestive enzymes complete the destruction of the pathogen. Eventually, that pathogen will more than likely then be released from the cell by exocytosis. That's what's going to happen if this is a neutrophil. If this is a macrophage or a dendritic cell, then we get an interesting another set of processes that occur where pieces of these degraded pathogens are reinserted on the surface of the cell, on the surface of that antigen-presenting cell. So we get some antigen presentation. We're not ready to talk about that quite yet. One other interesting thing that neutrophils can do as kind of a last breath attempt to localize infection is that once a neutrophil has phagocytized a whole bunch of pathogens, sometimes it will be stimulated to go into a self-destruct mode, and the cell will lyse, and particularly what it will do is it will dump its DNA out into the external environment and form this extracellular net. So this represents DNA out here that has been released by the dying neutrophil, and that DNA traps pathogens, kind of like a clot does almost, and keeps them localized. So that's called a net. So those phagocytic cells, particularly the neutrophils, are really pretty remarkable in terms of being able to be early defenders against infection. Okay, next week we're going to talk about 3 remaining parts of the second line of defense. We will talk about interferon, we will talk about the complement system, and we will talk about inflammation. Now remember, we've typically been having exams on Monday. The next exam is scheduled for a week from Thursday, a week from today, right?